about networking with people for job opportunities. There's the dreaming. There's falling asleep during conferences, although I'm pretty sure no one is falling asleep right now. And all of these are communications between different neurons. So this isn't just one single neuron that's firing. It's a community of different neurons that are interacting with each other. And each of them are making connections with each other. And these connections are called synapses. So a human brain would have billions of neurons, and there's 1 trillion of connections. So this is something that's very hard to study, but is essential for understanding how we learn, how we remember stuff. What we do in our lab is we try to understand neural connectivity, transmission, and plasticity. Even throughout our development, our brains are very plastic. They change. They're amenable to environment and different situations that you're in. And what we do is we try to see what are these communities doing together to do an entire goal. We see if they're modifiable by experience or not. And one of the things that we're interested in is this connection over here, at least in my project, is this connection between one neuron with the other and the synapse over here, and what happens when you have different changes to genes or proteins that are upstream to this. Now this is really important because the plasticity of your functional neurons is what makes you. These are some of the things that tell you what kind of intellectual capabilities are you going to have. And when you look into diseases, one of these synapses are the ones that are affected first. Now there are many players to it as well. There are intracellular communication, so communication between this cell over here and this cell over here. This cell telling this cell to pass the information through. There are secondary messengers, so there are neurotransmitters that are in there as well, something that you've learned from your basic biology courses. There are molecules that make them stick together, so these are cell adhesion molecules, make sure that these connections are always maintained. And then there's transcription factors. These are critical proteins that tell when genes are going to be expressed and at what time. So that's really critical in terms of understanding synapse formation and maintenance. Hi, and here's my PI over here as well, Dan Miranda. And one of the transcription factors that we study in our lab is called Daughterless. Now this came off from a screen that we did, Dan did in his postdoc, and this is one of the genes that I was interested in. This is a basic helix-loop helix transcription factor, which means that there are 2 helices that need to bind together, and then with the formation of this bond, they can bind to the DNA as well. So there are specific sequences that they bind to. And once they bind, they can either repress or activate different genes downstream as well. And Daughterless is the only type 1 basic helix-loop helix protein. In humans, there are 4 different types of basic helix-loop helix. So if you were to modify one of them, there would be another one that would come in, and because they're redundant, make sure that you don't see a phenotype there. With Daughterless, if you manipulate one of these genes, if you manipulate Daughterless, you can see the phenotypes much more easily. And it is also broadly expressed. Daughterless is also involved in 4 different things. It's involved in the formation of new neurons. It's involved in the formation of muscle cells. It's involved in sex determination. The reason it's called Daughterless is because these flies, the moms, don't have any daughters. That's why it's called Daughterless. So fly names are much more easier to understand. It's also involved in formation of the gut. Now all of these are very early on symptoms. And one of the reasons is because Daughterless is lethal. If you mutate this gene, this protein, it's lethal in early development. So that's one of the drawbacks as well. So the first thing that we did was we looked at homologs of Daughterless. So we looked at this protein called TCF4, which is found in humans and has similar function. And we saw that through different publications that it's important in brain development. It's involved in this disease called Pitt-Hopkins syndrome, which is a severe mental retardation. When you have a mutation in TCF4 that leads to this protein not being functional, will happen. One part of it not being functional, which leads to these symptoms that are severe. It's also involved in an age-related corneal dystrophy. called Fuchs corneal dystrophy, where the cells in your cornea start deteriorating with age. And what they found was that there's genetic variations in the coding region, non-coding regions of TCF4 that lead to this as well. So there's some correlation to it being important in post-embryonic development. So this is age-related. Also, risk of schizophrenia increases with mutations in TCF4. So clearly, there's something that's going on with neurons, the connections between them that is being affected, and that leads to problems. So understanding this protein's function is important in development. Why study Drosophila? And what I want to do is I want to convince you that Drosophila is a good model. I want to turn everyone into a fly biologist today, at least. That's my hope. But at least if I can't do that, I'll at least give you as much information as possible about it. The reason we study Drosophila is because it's small. It's easy to maintain. It's easy to raise. Their reproduction is quick. It's 12 days from egg to an adult. You can go from an adult that would lay an egg, that would go through this larval stage, to a prepupa, a pupa, and then turn into an adult. There's different range of phenotypes, so it's starting from the wing defects to behavioral defects. Those are some things that you can easily count, quantify. They're easy to meet, all right? Which, I mean, in other species would be a problem. And then their genomes are easily have been sequenced as well. There's homology between proteins in flies with humans as well. So the only drawback with this is that you have to turn over a process a lot. So you have to, these are always in vials, and you have to turn over these vials always, all the time. So the first things that we did was we tried to see the localization of Daughterless in post-mitotic neurons. So these are after embryogenesis. There were a previous study done in '93 by Cronin and her colleagues that saw that Daughterless was expressed throughout, but there was a strong expression in the lobes. These are the optic lobes. These are where the neurons from the eye would come in. And there was non-uniform staining in the ventral nerve cord as well. So what we did was we looked at the same thing using confocal microscopy. And what I'm showing here is a marker called ELAB, which is a marker for post-mitotic neurons. And then we see the expression of Daughterless in green. Now, one of the things that you'll notice is that this protein is expressed in axons. So these are bundles of axons that are coming in from the eye. These are connections between the two lobes. You see them at the photoreceptor axons. These are axons coming in from the eye. And the ventral nerve cord, which is involved in locomotion. So these are expressed after embryogenesis. So if this was an important gene for forming young neurons, what is it doing after those neurons are formed? If it is a transcription factor which would be found in the nucleus, what is it doing outside the nucleus? So these are some of the questions that I am answering. And what I'm showing here is what we did was we imaged it throughout development. So this is a brain of a larvae, and this is a brain of an adult. And this is something that's maintained throughout. So one of the questions I had was, does Daughterless have a post-mitotic neuron function? And again, these mutants are lethal, so that's why it wasn't easy to understand what was going on in them. And we wanted to study this in a spatiotemporal manner, so at a specific place at a specific time. And we wanted to use some very well-defined circuits to study this. To do this, what you do is use a GAL4-US system, which is a bipartite system. Flies do not normally have GAL4s. These are proteins that are expressed in yeast. So what we did, what geneticists do is they put it into a fly, and then they can turn on or turn off gene expression. So there are three different combinations you can have. You can have a combination of US GFP with plus, a plus with plus. There wouldn't be anything going on. The other case could be where you have a GAL4 over plus, and this is only going to be expressed in these regions, but there's no expression yet. When you put in a GAL4 with a GFP, this protein comes and binds to the upstream activating sequence, producing GFP, and what you can see here is only these tissues are expressing GFP. What we did was we used something called an RNAi interference to knock down Daughterless. So if you study this protein, you would need to cut it to reduce its expression and see what the effects are. And what you do is you degrade the mRNA target. So we used a specific Daughterless target RNAi, we degraded it, and it came to be about a 60% knockdown. We also studied the neuromuscular junction. And what it is that you have an axon that comes to the muscle, makes connections, and when you look closely to it, you can see different neurotransmitters that bind to these dots called the active zone. And you have the muscle fiber over here that has the postsynaptic densities. Overall, what we saw, what we could do is we can knock down Daughterless expression or overexpress it. So what I'm showing here is Daughterless expression at the synapse. So this is Daughterless antibody that we're recognizing in the muscle nuclei and the axon and the synapse. This is really important because what we're seeing is we're seeing it after a certain development, and we're seeing it outside the nucleus. When we knocked it down, what you can see here is that we can reduce it in the axons and in the synapse as well. We also looked at their Bouton structure. So what these innervations do are they make these really neat button-like structures on the muscle. And what you can do is you can count them according to their sizes and the total number of branching. So what we see is that there's increased number of these boutons, these innervations. There's also an increased number of branches. So they're forming more branches, they're forming bigger boutons. We also looked at these docking centers called the active zone, and we see that there was a reduction with a knockdown of Daughterless. So the two functions that we see is that there's a reduced number of active zones. We show that there's an increased number of Bouton and branching. So we wanted to see a functional role of it as well. And what I'm going to show really quickly is what we did was with behavior. So what we looked at was we looked at a larvae's number of contractions. So a larva would move in a spatiotemporal manner. It would move upwards by pushing its muscles in a stereotypical manner. We counted the number of contractions they had and the total distance that they crawled. With knockdowns, they had fewer number of contractions. With knockdowns, again, they also traveled a fewer distance, but it's not as drastic as the number of contractions. So what we see is that with contractions, there's less number of with knockdowns, there's less number of contractions and there's less distance traveled as well. So this is what our hypothesis is that when you knock down Daughterless in a post-mitotic neuron, after embryogenesis has happened, they have a function there. They have a function in the number of branches that you make and the number of boutons that you make. And that being said, that's all I want to keep on for today because I'm over time. Thank you, everyone.
Our second speaker for today comes from a different discipline. Steve Essinger comes to us from the Department of Electrical Engineering in the College of Engineering. However, he has a biological slant to his research in keeping with, I think, the general biology theme that's going on today. Steve is developing methods that can be used to look at species interactions and to start to develop hypotheses from these interactions. Steve, take it away.
All right, thank you. First, I'd like to thank the GSA for inviting me here today to give this talk about my research experiences here at Drexel University. I'm actually a product of Drexel Engineering's undergraduate program. I graduated in 2006, and I went and took a job with a company called Metrologic when they were my third co-op, and I took a full-time job with them. And with this was a team I was working with in Suzhou, China. And we were developing this electronic paper display. It was this bistable material that you could update the information content. You know, you could display a price or a piece of information, but it only required power to change the display. So you got really long battery lives. You could run this thing for 5 years separate. So it was some pretty cool technology that we were working with. And we wrapped this wireless network around it. And so that the idea was that you could just take these signs and put them through like a retail store, and then from a central database in the back, update all the prices as the price of the product would change. So this was really cool technology. I had a lot of fun. I got to do the systems. I got to do hardware, software. But it was only one part of electrical engineering, or engineering in general. And I knew I wanted a career in engineering, but I also wanted to look at the other side, which was the signal processing, the mathematical modeling. So I decided when I went back to grad school that that's what I was going to focus on. And furthermore, I decided that I wanted to do something interdisciplinary. I didn't want to just necessarily work with just electrical engineering, technology, and traditional products, but to branch out. So I met Gail Rosen, and she had a bioinformatics lab that's now called the Ecological and Evolutionary Signal Processing Informatics Lab, also known as the EASY Lab. And I thought this was really cool because Gail was in signal processing, but her focus and the application domain was towards the biological sciences. So this sort of gave that interdisciplinary that I was looking for. Now, traditionally in our lab, let's see if I can get the laser point here. So we're working with biologists who are in the wet lab. They're doing the data collection. They're working with the cells. They're sending that data out. And this is a picture here of some gene expression data. It could be DNA sequencing. And it's really the data that we get out of here that we're most dealing with our modeling. And it's mapping it to numbers. We're looking for patterns within this data. We're looking to communicate information that we find so that biologists can make decisions on their data and move forward with their research. So when I was working in the lab, Gail introduced me to Jake Russell. He's in the biology department, molecular ecology, and Jake was, he was the domain expert, right? He's the one that had the focus. And he had introduced me to the wonderful world of bacteria, which I really had no idea about. But through Jake, I had learned that bacteria are the most abundant organisms on the planet. Right, so they're found everywhere. Soil, water, they're in the air. I had just recently read that hurricanes actually can bring populations of bacteria through that were not there before. So I just found that to be really fascinating. You know, a handful of soil has billions of cells in there, so they're incredibly abundant. The human body harbors about 10 times more bacterial cells than we do human cells, so I found that to be very fascinating. They're incredibly diverse. There's over 500 different types of bacteria species within our guts. So given all of this, it was clear to me that bacteria are very important. There's definitely a relationship between human beings and bacteria and the environment, and it is something that has been studied and is going to be studied for a long time. So I thought it was a very cool application domain. Now, when you're dealing with bacteria, traditionally, you have single cell analysis where you would have one type of bacteria that would grow in the lab, and you extract the DNA from it, and then whatever information you're looking at in that DNA, you know, barring any contamination, belongs to that bacteria. The problem is, though, is that only about 1% of bacteria can be analyzed in this matter. So what we have to do is, if we want to study bacteria from the environment, we have to take samples of them using the field of metagenomics, in which we take a scoop of soil, for instance, and we sequence the DNA of the entire metagenome. And we get this spaghetti mess of DNA that comes out of it. And then the DNA sequencer is going to give us different fragments, different pieces. Now these pieces can belong to different organisms, they can belong to the same organism, but from different parts of the genome. And the question becomes is, how do we classify those fragments? What do they belong to? And can we assess some sort of measure of diversity about the community sample that, from which these bacteria originate from? Now, at this time, Gail had introduced me to Roby Pulakar, and he does machine learning at Rowan University. Now, classification is bread and butter for the field of machine learning. So I work with Roby and Gail, and we work with some supervised classification where we have a known reference database of sequences, and we're trying to take these fragments and classify them to that database. And there are many popular tools for that. And we get reads that come out, we're able to do some identification. And then we filter the reads, and then we have certain clusters, so we know that all these DNA fragments come from, let's say, this species A, species B, species C. But then there's a bunch that we don't know about. And those, we would at least like to get some sort of measure of where these bacteria, like what is the diversity, within that sample. So we do unsupervised learning, which is when you don't have a reference database and you have to make sense or find patterns within your data just using the inherent information content that are in the DNA sequences. But supervised classification is very popular. Most biologists will use it anytime you have to classify a DNA sequence. So we did some benchmarking of one of the popular tools. This would be BLAST, and most biologists would be familiar with this. And we wanted to see how changing different parameters that the algorithm would use, how does that affect our results. So BLAST is supervised, so we have a training database or a reference database from which we're referencing our query, those DNA fragments to. So we put that into a whole, the whole genome database here, and we can try different things. We can break it up, whole genomes, we can take parts of the genomes, different sizes, right, and have that as our reference, and then keep some of that information removed from the algorithm so that it does not have reference information in that. And then also on the input, we have the fragments. And we can introduce different types of error because sequencing technology is not perfect. We can introduce mutations into that. And so using these different input scenarios, we can run the algorithm and then we get output different performance measures. And we can get a sense of how these algorithms are performing and identify areas for improvement or best practices. And typically we get like a plot like this out, and in the interest of time, I'm not going to go over this, but again, we get some data that comes out and we're trying to make recommendations for how best to use these algorithms, or perhaps what algorithm would be best in a given scenario. So at this point, I was wrapping up this project and Jake introduced me to Karen Sullum. She was a graduate student in the biology department. And Karen is interested in studying the fish, the gut contents of fish, the bacterial communities. And so for this project, we did a meta-analysis where we were looking at, we combed the web at many different databases where DNA sequences that had been submitted that were obtained from different fish gut from all over the world. And we assembled these, and Karen had some questions that she was looking to answer. She had some hypotheses that fish will have similar gut microbe content when they have similar feeding ecology, the fish are more closely related, or the fish are from the same habitats. And so we went through and we used the classification algorithm I just showed before that we had benchmarked. And we wanted to get a measure of how similar different fish guts were to one another. Well, as you do the classification, you still get many different classifications of bacteria that come out of this, and to look at it on a piece of paper or a spreadsheet, it's just mind-boggling. So we need methods to be able to reduce the dimensionality of this data from very high to low and have some sort of way of interpreting. And it's much easier to do this when you have visualization. So we take the high-dimensional data and we project it down, and we group these based on samples. So like these teal markers here are all from fish from a natural environment. Then we have other fish here that are from environment, but they're cultured. So we see that cultured and natural environment are separate from one another. These black ones over here are gut microbes from other vertebrate guts, non-fish. So we see that there is definitely separation between the gut contents of the organism and where the organism had originated from. And so the conclusions of this study were that there was more specialized gut communities that were previously recognized. And it raised further questions like, do these gut microbes, do they have further adaptive roles? So do they adapt based on the change in habitat? And how stable are these associations within species? So Karen went on to study this in more detail, and she went to Trinidad and studied guppies. And I started thinking more about how do bacteria and species respond to different environmental conditions. So at this point, Gail had introduced me to Chris Blackwood, who's a molecular ecologist. He is at Kent State University. And Chris and I, we were looking at modeling species responses. So we're looking at, let's say we have a piece of land here, and across this land we have decreasing precipitation. So we call this change in environmental condition, environmental gradient. And we sample at each one of these points here. We look at the take a scoop of soil and look at the metagenomic content classification. We look at the bacteria that are there, and we can assemble those in some sort of matrix where you have species as the rows and the columns correspond to different sample sites. So if we look across the rows and plot them, we can see how are the species responding to a change in this environmental condition as we move across here. So we started looking at these curves, and we wondered if there were some models for how species respond in general to environmental conditions. And we found that generally you can use a unimodal response curve, where you have the mean of the response would be the preferred condition for a given organism, and the tolerance, being the variance, is how sensitive that organism is to environmental condition. So the x-axis is the environmental gradient, pH. It could be pH, soil, pH, salinity, temperature, precipitation, and then the y-axis would be the abundance. So species, this red species here has a much, whatever this gradient is, let's call it pH, it has a much less, it's very sensitive as opposed to the blue, which almost isn't affected by the environmental gradient. So we had a species model for how they were responding to the gradient. So then we started thinking, okay, well, what about bacteria responding with one another, right? Organisms respond to abiotic, right, physical factors, and then there's also biotic, which is the interaction between different organisms. So what we did was we set up to develop a detection algorithm to model biotic covariation, and we want to detect if this is going on once we have accounted for these different environmental conditions. So we use a species response model, and we were just using a simple linear model, and we're using a multivariate regression, and we're using it as the explanatory variable would be the pH or the salinity, and we would use that information to remove that response from the overall species response, and then we would look at the residuals of that. And within the residuals, we would do a hypothesis test and look for significance of covariation. And if we did find it, we could make a decision on whether there was covariation among those samples and those organisms or not. So here's one plot of looking at residuals. It's a bunch of garbled. These are species responses here across sites. And looking at it, we don't see much covariation in this case. And these leftover abundances, after we have removed the environmental gradient, is due to noise, could be arise from measurement error, perhaps a gradient that we had not removed from the response that we didn't cover. But over here, though, we can see that whenever the blue species goes up, we see that the green species goes down. So we have a negative correlation or negative covariance among these species that we can visually see, but can we quantify it, though? And so we look at those two species, and we look at pretty much all the species within the community in a pairwise fashion, and we pass their abundances through a randomization model that's specific to the covariation of species within the environment. We call this a null model. And we generate many of these randomized matrices, and we determine whether the covariance of the original is significantly higher than the ones of those randomized. And if it is, then we can stay with some level of statistical significance that there is covariation in these samples. I'm just going to skip that in interest of time. So we have a model now that we're pretty excited about that works well for detecting covariation among species. So now we want to apply it to real data. There's a study here that was in the Baltic Sea of a salinity gradient. So the environmental condition is changing the salt content. And from the study, we know that salt is definitely structuring this community. However, now we can take that salt vector and remove that from all the species abundances and then look to see if we can find embedded covariation among organisms within that. So just to wrap it up, I was asked just to talk about where things are headed, and I found two quotes here that I thought would be interesting, to keep in mind. And I'll paraphrase Wayne Gretzky, try to never skate to where the puck is, but where it's going to be. Right? So we want to look at where things are headed and try to use look at an idea of what is going on in the field right now to kind of project where things are going. But on the flip side, though, we know prediction is difficult, especially if it's about the future, as Niels Bohr said, who is somewhat of a pessimist. So we look at the cost per megabases of DNA sequences. This is from genome.gov. And from 2001 to 2008, the cost of DNA sequencing has decreased with approximately at the same rate of Moore's Law. Any technology that keeps pace of Moore's Law is generally considered to be very good technology. Around 2008, it dropped substantially and continues to down in 2012. And as we look in the future, I just read an article that there is a huge DNA sequencing facility being built in China right now for the commercialization of sequencing human genomes. And they believe very shortly, within the next year, and I know they've been saying for a while, but I believe they're going to hit it soon, that it's going to be a $1,000 genome, and shortly thereafter that, it's going to be in the hundreds of dollars to sequence a human genome. So cost is very quickly going to become irrelevant for collecting this data. So we're going to be able to enable a lot more experiments and data collection. Secondly, if we look at the percentage of the science, technology, engineering, mathematics jobs in the area through 2018, we see that 71% are expected to come from computing. So we have people that are going to be highly skilled in programming, but also computation and analytics. So if you couple that with the decrease in sequencing costs, I think we're going to be able to move through a lot more data than we have been able to over the past decade. And I think what's going to be important is to collect as much data as possible and then use analytics to determine what is useful and what isn't. Because it's a cycle, right? So as we find more information based on our study, it's going to prompt more biological experiments, and we're going to perhaps want to go back and look at that data. So I'm a proponent for sequencing everything and making sure it's in the public databases so that it's accessible to everyone. And that's it. Thank you.
To introduce our third speaker. The third speaker brings the focus of interest back to the Department of Biology, keeping with our majorly biological theme. Our third speaker is Cronin, who works with Dr. Gail Hearn on her conservation biology research. Drew's work takes place in the field in Bioko Island of Equatorial Guinea, and he's probably going to have the coolest and most scenic pictures of the three speakers. I always say that Gail's lab has an automatic advantage because of all of their images. Drew is going to tell us a lot about his research, which has focused on the effects of bushmeat hunting on the primate population on Bioko Island. Drew, you're up.
Hi, I'm Drew Cronin, Dr. Gail Hearn's lab. I'm going to talk about the impact of hunting on primates. And just to give you a little bit of background, I'm going to come back to this idea of how I spent half of my life in graduate school living in a tent. Very different from most people here, especially at Drexel. So how I got here, I'm originally from the Jersey Shore, which most of you know like this. I know it as this, so I spent my life on the beach, in the forest. Some might say living a very good life, but there are tourists, so I do deal with this. But I developed a real love for the natural world and getting my hands dirty, and then eventually what happened was I went to New Zealand to study abroad as an undergraduate, and I just fell in love with the place. The natural world is amazing. And I was really taken by the work that the Department of Conservation does there. Right? So it's a small island or set of islands, and they deal with a lot of environmental problems, but they've got the population, for the most part of New Zealand, to buy into this idea that conservation for prosperity, right? Investment in natural resources is an investment in your future. So, you know, I really dug that. That's sort of where I'm at. And what happened was I came home, I took another opportunity to go abroad, and that took me to Bioko Island, Equatorial Guinea, which is where I've been working since 2005. Little island off the coast of Cameroon. It's made up of 3 volcanoes. It's very, very small. Just above the equator, this area down here in the south gets over 10 meters, so over 30 feet of rain per year. The capital city up here in the north has approximately 100,000 people, but it's a developing country, so census isn't very good. And then there's these protected areas, which you should just remember, making about 40% of the island, but there's no guards. So I go there, I see it, wow, it's just like New Zealand, lots of forests, lots of cool mountains, good stuff. They've got these beautiful monkeys, which I'm going to talking a lot about today. So lots of great monkeys in the forest, right? But then you still have the conflict, right? You still have that environmental degradation that you need to work against, right? So this is a drill monkey. It's the largest primate on the island, being sold for consumption in the market in Malabo, in the capital, right? So that's bushmeat, right? It's a commonly used term for this type of thing. So it's any wild meat that's hunted and sold for consumption, right? Highly valuable as a non-timber forest project, so not wood product in the tropics. It's really commercial. In the past, it was believed that it was a subsistence thing. It was local people in the villages, and that does still exist. But for the most part, it's a commercialized venture right now. In many areas of the world, especially in the tropics, it surpasses habitat loss as the primary threat to the large invertebrates. And in West Central Africa, where I work, it's thought that over 60% of the mammalian taxa are harvested unsustainably. So you've got really, really large numbers of animals coming out of the forest. And what drives that is high value relative to the cost of doing it. So all you need is a gun and a shotgun shell, which is not too much investment. And then by the end of the year, you can be making a lot of money. And what drives that is the growing urban demand. So the economy in Africa is growing. People in the cities have more money, but they still have their cultural heritage, and they've grown up with eating animals from the forest. And so the wealthy elite in a lot of these cities are buying the bushmeat, and that is driving people out to the forest to supply that. Okay? So Bioko, I call it a bushmeat case study because it's an island, it's a contained system for a number of reasons I'm going to go through. But more importantly, commercial hunting is really the only threat to any of the large invertebrates on the island. It's a luxury item, so you can go into any supermarket and get protein for significantly less than anything that you could buy in the bushmeat market. Even a rat. I don't know if any of you guys are keen on eating rats. It costs about $10. All right, so you can go get a chicken for about half that cost. All right, now, for other reasons, it's a case study in that, like I said, it's a contained system, right? It's very small. For any of you that are familiar with Jersey, again, best state in the union. This is the Pinelands Management System. That's about double the size of the island I work on. The consumption is largely restricted to the capital in Malabo. There's only two major routes to the market. So on the mainland of Africa, you've got a really convoluted market structure coming from many different places. But here we've got two roads that are the main things because there's a big, giant mountain right here. They can't really drive through it. The market's almost entirely for profit. So like I said, most of the consumption is in the capital. And most of the hunters are non-native. So they're not the indigenous tribe from the island. And they're from other countries, not just Equatorial Guinea, but from Cameroon and Nigeria. Like I said, it's a steep place. Deforestation's not really having any impact, not yet. But a lot of the places you can't get trees out of, so you've still got intact forest. And then it's not fulfilling the economic or dietary needs of the local population. Offshore petroleum makes up a whole heck of its economy. The bushmeat economy here is a fraction of 1%. And then when you talk about the dietary needs, the yellow circle here is an estimate of the minimum protein requirement for 100,000 people. And this is all the protein that we estimate coming from the market, and this is all the protein that is coming from primates in the market. So it's not people doing this to survive, right? So what I wanted to ask you, or what I wanted to study on Bioko is how you quantify hunting. How do you quantify essentially an illegal activity? And then what are its effects on the anthropoid primate? So how do we get from the wild population to the changing populations to a dead monkey in the market and then eventually the dinner table? Right, so I have three major goals, and it's to assess the temporal trends in the market, to try to understand the market structure, to understand the wild populations, and then to see if maybe it's just the ecology. Maybe we're just totally blowing this thing out of the water. So what I'm going to talk about today is predominantly the temporal trends. All right? So we've had somebody that we employ do market surveys since 1997, and I'm just going to focus up until September 2010. And they go into the market 6 out of 7 days per week, and they take a whole suite of data on all the carcasses that come, and then we break that up into certain aggregations. So by taxa, so avian, primate, reptile, rodent, others, essentially non-primate, non-rodent. The capture method is either traps, snares, that's sort of lumped together, shotgun, and then other, sometimes things that got hit by a car. But predominantly trap or shotgun, and then geographic origin, so whether it's from the island or it's imported, so from the mainland.